Okay. Good morning, ladies and gentlemen. Good morning, panelists, and good morning, participants. I would like to welcome all of us to this very important webinar that is being organized by the Nigerian Academy of Science. The theme is Variants and Waves of COVID-19 in Nigeria, to be or not to be. We have our draft agenda. We will start. I'm not sure the executive secretary has joined. If he hasn't, then we're going to skip his part and then we'll move over to the president of the Nigerian Academy of Science, Professor Ekabrede, FAS, for her opening remark. Professor Ekabrede is unavoidably absent, and so we're going to share her welcome address. So that's my president, Professor Ekabrede, FAS. It's going to be a video. While we're waiting for the video to play, I would like to inform the participants, if you have, after presentation, if you have any questions or clarifications you want to be made, just use the chat box. We'll address all of that at the end of the presentations. Adamu Salahuddin, we'd like to welcome you.
Prof, good morning, sir.
Morning, welcome.
Thank you.
My executive secretary is here.
Thank you, sir. It's just
you're welcome.
Thank you, sir. We've been having some Zoom issues, so the link wasn't coming through to me.
Yeah, that's fine. You know, Lagos, the hold up is in everything, including the internet.
I've also joined now, so I can give my address after doing speaks.
Yes. My president, you're welcome, ma.
Thank you.
Yes. Okay, ma. So with the directive, just go ahead and welcome everybody to the fourth COVID-19 webinar. We've had several webinars, but this is the first purely NAS COVID-19 webinar. We've also had others that were regional in collaboration with other academies in Africa and even globally. But the academy itself has had 4 webinars. This is the first one focused on Nigeria, talking about the issues that might be peculiar to us. And this is particularly crucial because we're looking at the variants. And I think the variants address the future of COVID-19, really. The issue of variants addresses that squarely. It points at issues like, would we be taking vaccines forever? Or would the virus mutate and die out and we'll be free once again? You know, so we have eminent experts to help us navigate this topic. But mine is also just to say that the academy is the right institution to put up this kind of discussions because this is the highest independent scientific body that we have in the country. And the academy was founded in 1977 It's the the oldest academy we have, and of course the most senior. And the academy aims to help the country address policy issues with the use of evidence. So we expect that this is the kind of issue that the academy would definitely intervene on, as we are doing. And we hope that today people would enjoy what we have and also help us to take the message forward. Particularly, we're hoping that here you will hear unbiased scientific opinions, as we definitely always try to do. And I'll say I'll invite you, in order not to take so much more time, I'll invite you to visit our website, which is www.nas.org.ng, for more information, and feel free to contact the Secretariat if you need any information. Lastly, I must thank Professor Uzochukwu, who's the moderator today, Professor B.S. Uzochukwu, FAS. And of course, now I hand over to the president of the academy, Professor Ekanembraid, FAS. Thank you.
Thank you very much, Secretary. Yeah. So over to you, President.
Thank you, Chairman. Distinguished ladies and gentlemen, I bring you greetings from fellows of the Nigerian Academy of Science, the foremost science body in Nigeria. In line with our mandates to assist in solving national problems through application of science, technology, and innovation, the Nigerian Academy of Science has been working closely with government and other stakeholders in the national response to COVID-19 pandemic. The Academy has issued a number of statements on COVID-19 and has conducted webinars on the pandemic. The first webinar, hosted on October 6, 2020, focused on non-pharmaceutical interventions in COVID-19 containment. The second webinar, hosted on October 7, 2020, was on essential health services amidst COVID-19 and future pandemics. The third webinar, on October 13th and 15th, was on COVID-19 towards the new normal. Some of these webinars were held in collaboration with other academies and funded by various bodies. This is the fourth in the series of webinars conducted by NAS, and the theme, Variants and Waves of COVID-19 in Nigeria, to be or not to be, is apt at this time, when variants are now posing serious problems in the response to the pandemic. The panelists will, of course, talk us through the basics of viral mutations, their significance, COVID-19 variants, and the implications for Nigeria's public health response. We are grateful, very grateful to Professor Christian Happi, Director, African Center of Excellence for Genomics and Infectious Diseases, and Professor Sunday Omilabu, Director, Center for Human and Zoonotic Virology, College of Medicine, University of Lagos, for accepting our invitation to enlighten us on tracking COVID-19 variants in Nigeria and managing variant-driven waves of COVID-19. We are also grateful to our fellow, Professor Benjamin Uzochukwu, FAS, for accepting to chair this event and for starting the event on the dot of 11. At the end of this webinar, we would have gained significant knowledge on how to manage the pandemic, which is now getting more complicated with the emergence of more COVID-19 variants. In all, Nigerian Academy of Science webinars on COVID-19 will provide reference information relevant to managing future epidemics and pandemics. I wish you all a pleasant experience at this webinar, and I thank you for listening. Over.
Thank you very much, my president, Professor Ekanembraid, FAS, for that wonderful opening remark. We're grateful we have you at the helms of affairs to direct this noble association. And I want to congratulate you for all you have been doing. Now we'll move over to the presentations. So today we'll be listening to very prominent professors in this area. They will be talking to us on various issues within the context of the different variants that are coming up. So the first in the list will be Professor Christian Happi. Professor Christian Happi is a professor of molecular biology and genomics, and he's the director of the World Bank funded African Center of Excellence for Genomics of Infectious Diseases in Redeemers University, Ede, Osun State, Nigeria. Professor Happi, in an unprecedented way, recently used next generation sequencing technology to perform the first sequence of the new SARS-CoV-2 in Africa within 48 hours of receiving sample of the first case in Nigeria. So this seminar work provided an insight into the detailed genetic map of the new coronavirus in Africa, not only confirming the origin of the virus, but also paving the way to the development of new countermeasures, including new diagnostics, therapeutics, and vaccines. So Professor Happi will be speaking to us on tracking COVID-19 variants in Nigeria. During his presentation, he will cover areas such as what virus variants are, how they happen, and why they occur, the COVID-19 variants, the current types, testing, and detection of variants. So ladies and gentlemen, join me in welcoming Professor Christian Happi. You're welcome, sir.
Thank you, sir, and thank you for the introduction. I want to thank the Academy for giving me the opportunity to present this work. And I will just go straight and then talk about, how we've been able to use genomic as a way to track the different variants of COVID-19 in Nigeria, but also thinking about, then talk about how we've been able to translate this knowledge, because we're talking about Academy of Science, how we're able to translate this knowledge of genomics and tracking these variants into countermeasures like diagnostics and effective and vaccines. And finally, we'll talk about, I will mention the capacity building component of it, because we don't want this knowledge to actually remain in our institution. We're building capacity not only in Nigeria, but also in Africa, in order to make, prepare not only Nigeria, but the rest of Africa, to preempt the next pandemic. And we use genomics, as a tool, really first demonstrated the usefulness and the power behind genomics when it comes to pandemic response by applying genomics to the Ebola response in West Africa. And we have demonstrated from that time that genome sequencing technologies has revolutionized the way we do research and also the way we can approach healthcare. And this really has tremendous benefit, not only into pandemic response, but also has a lot in terms of technological advancement and also health benefit, and also to promote and boost our ability as Africans, how we can respond to diseases. Now, specifically for COVID-19, when the first case of COVID happened in Nigeria, that was around March last year, we received a sample from the Nigeria Center for Disease Control in order to reconfirm and then characterize this virus. And the reason being the fact that the Nigeria Center for Disease Control knew about the capacity and then the power behind the technology that we have developed, and also in terms of infrastructure that we have on site. So what we did at the time, and then the secret behind it, and why we did something that was unprecedented, is the fact that we pulled out the big guns. We have a system that is fully automated in our lab, as you see here, automation from extraction to having the largest sequencing facility or the best sequencing facility or platform in Africa. And using a platform such as the one I'm showing here, which is the NovaSeq 6000, that it actually has a very powerful throughput, but also quick turnaround time. And we were able to do something that was unprecedented, and that no other lab or country in the world have achieved that is basically sequencing the genome of the first coronavirus in Nigeria within 48 hours of receiving the sample. But then we were able to really pinpoint, exactly, with laser accuracy that although this patient was from Italy, he got infected in Switzerland, as you see here, between these two sequence data, and then travel to Nigeria. And then this is where, and then effectively was a first case in country. We demonstrated also that at that point, there was no community transmission. It was an imported cases, as we demonstrated in other African countries that we've been supporting through this outbreak. So, and that was the first COVID-19 case, not only in Nigeria, but the first in Africa, and then the fastest in the world in terms of turnaround time, 48 hours. And we've continued to support Nigerian government in responding to COVID-19 by providing information almost in real time, using sequence data and giving information. Here you could see, for instance, areas where we've covered. We've been able to generate, and now this data is a bit obsolete, over 1,000 genome in Nigeria, but we've been able to cover about 30 states, of the Federation. And that really has been able to provide information. As you can see here, we knew over the time what happened to different states, the different variants circulating in all of these different states. We also knew with accuracy, as of today, for instance, that we have over 48 different variants circulating in Nigeria and in different states. And these information are often given to the presidential steering committee for COVID-19. We do this, and we'll give this update every Friday or Saturday, and then they meet, and then the decision that you see in these countries are often taken based on the results and the data that we provide them. Here, for instance, you can see here how we were able to characterize this virus, and then this dashboard that is available that we designed that is available to the Nigeria Center for Disease Control. You see this variant evolving over space and time across the country, based on the sequence data that we generate. And these informations are very crucial because they provide almost real-time data, and the decisions in the country are guided by science. And there's reason why you are hearing the former director of NCDC, Dr. Chikwe, saying that, look, science guides the response in Nigeria, basically because we provide this information very quietly in the background, and then they use this information as a way to formulate the necessary policies that are available. As I mentioned before, we know exactly the different lineages. We have over 45 different lineages of variants circulating in Nigeria. And then among them, there are variants of concerns, variants under investigation, and then variants of interest. I will talk about that later on. But one thing that we also did, again, when we talk about tracking the variant, is actually during the lockdown, we were able to use genomic information to monitor the way variants were moving from state to states. And then this is a case, for instance, where you look at Edo State that was in the 2020s, or your state, Osun State, Lagos State, FCT and Akwa Ibom. And if you look at this diagram, you will see that at some point early in the pandemic, this particular purple variant was not present in Edo State, but then you can see it increasing over time. And you also see, your state that wasn't present, but it pops up at some point toward the end of May. And you see that moving along the different states. And by the time we tracked this particular variant, we realized it was a variant actually that was moving from Lagos to the rest of the country. And this information, we provided this in the background, to the government. And then they were able to see that despite the lockdown, you could see variants moving from state to state. And this was basically due to the fact that during the day, the law enforcement officers pretended that they were enforcing the laws and people were not moving. But at night, we realized that, they were allowing people to move and moving along with the variant. And that's why you could see variant moving from state to state across that time. This information was shared with government, and then they made the right decision in terms of reinforcing the lockdown and then creating additional barriers. But then this is the very first variant, which is mutation that was observed in the ancestral variant, which is a lineage A from Wuhan. And this D614G mutation, we were among the first group in the world to identify that, and show that this variant was associated with increased transmissibility, as you see here, demonstrated in this map. Then in vitro analysis actually confirmed our suggestion at the time, and that really was the very first point mutation demonstrated to be associated with increased transmissibility, and this was during the first wave. When Nigeria faced the second wave, there was an unprecedented number of increase of cases between November and December and January, 2020, between November 2020 and January 2021. Then you could see here among the state where you have all these major airports, you could see the spike around these areas. And this was really due to the fact that travel could actually move these viruses from place to place. So we asked the question at the time, what was responsible for this, for the spike that we're observing in Nigeria? At that point, people were talking a lot about the UK variant, or what they call the Alpha variant right now. But what we found in Nigeria was a bit different from the Alpha variant. Apart from the Alpha variant, we observed that in Nigeria, there were some other variants that were driving this transmission. And one of such variants that we discovered at the time was a B11, which is now called the Eta variant, that emerged somewhere around Edo State early in December, and then was also driving the transmission. This variant share a lot of similarities with the Alpha, the Beta, and also the Gamma variant, in the fact that they share a lot of mutation in the spike protein, and this variant has been associated with also potential of immune escape and vaccine escape. This variant actually emerged from Nigeria, spread across, and then seems to be the largest second variant in Africa now after the Beta variant that emerged in South Africa. Tracking this into the country, we know exactly, as you see in the map here, where we see this variant based on sample that we receive, and these information are also shared with government. We also discovered another variant called the B11318, and this is a variant under investigation that emerged from Nigeria and then spread in many countries in Africa. This particular variant at some point created a lot of havoc in Mauritius, whereby it was responsible for about 90% of the cases of SARS-CoV-2 in Mauritius. And that really also has a strong it has a lot of mutation on the spike protein, and also has potential for immune escape or vaccine escape. Then recently we identify almost at the same time with the Nigeria Center for Disease Control, the Delta variant in Nigeria, and we'll be monitoring this variant. And this is characterized by SNPs in the spike protein, especially, the T19R, the L452R, the T478K, and then the P61R mutations. These mutations are actually in the pocket that has the potential to interact, with antibodies and then those really facilitate, makes this variant a bit difficult, for the vaccine or immune system. So these are basic mutations that are characterizing this variant. But then what we go on is basically showing here, how we've been mapping this over time and looking at different mutations that are accumulating on the variant that are circulating in Nigeria. And what you see here are basically these mutations that are very, very important in the spike protein, and then that could be involved in immune escape and then vaccine and also vaccine escape. And over time, as you can see here, you could see, almost real time, month by month, and then depending how the dynamics of variant in the country, and you could see all those different variants here. These are the major ones. And you could see here, for instance, how gradually in the month of July, the Delta variant is becoming the most dominating variant in the country. But then this has also allowed us to actually look into the vaccine, for people that had vaccination and then that came down with infection. We reported the very first case of vaccine breakthroughs in Nigeria, but then this is through the Presidential Steering Committee. And then this is the first case whereby they reported that case. This was a cluster in a family of people, of a grandmother, a 2-year-old baby, and then a husband and wife. And these are people that got vaccinated and then got this breakthrough infection 6 weeks after completing their second dose of the AstraZeneca vaccine. And this really was very important. We did not only just sequencing, but we also look at the immune profile of this patient. And the Delta variant was actually the one that broke through, this infection from people that were vaccinated and completed their vaccines. Now, the second part, or the next part of this is how we translated this information. What do we do? All of the genomic sequence data and things that we generate. We not only just use the sequence to actually help public health response, we're also translating them into point of care diagnostics, pivoting from the point of care diagnostics or the CRISPR, which is gene editing technology that we actually used to develop the first gene editing technology for Lassa fever. And this article published in Nature actually described clearly how we were doing this in our lab, but we actually just borrow from this concept or for this novel concept that we developed a few years, 2 years ago, and applied that to CRISPR, applied that CRISPR technology to COVID. So we went on to develop the very first COVID CRISPR-based diagnostic that can enable us to have a diagnosis for COVID, within less than an hour. So, and then these are room temperature without requiring any equipment. And this point of care diagnosis, like the pregnancy test, can be done by anybody. You don't need to be, educated to do it. But we also have a version that can increase the throughput. We do them into plates, and then you can do 96 tests at a time. This test was highly reported in the NBC, the US, for instance, and then TV5, and then the BBC. And then this test has been approved by the US FDA, and then is used in the US, and it's also been undergoing clinical trial in some other, in some other places in Africa. This basically just giving you the first test that we did in Africa, in Nigeria, the first 500 tests. Very sensitive, 98% sensitivity compared to PCR, 100% specificity. But then we were able to develop this test within 2 months of the outbreak. But what is important here is actually we've been able to demonstrate through this pandemic that we can actually develop new tests, within a week of this, getting a new virus or understanding the new virus. We're also multiplexing this test, meaning that, we can bring this test together with Lassa fever, malaria, and others. And this is nucleic acid-based test into a new test that we call common. This technology has also been, we published this in Nature, and then the previous ones are also published in Nature, demonstrating, how reliable this technology is. But we've used information also, as I said before, getting information, sample from people that had breakthrough infections, to develop what we call an antibody neutralization assays. And this neutralization assay will tell us how viable are the antibody responses from people that got vaccinated, and then to people that had been natural survivors of this virus. And this really is a very clear demonstration using the antibody neutralization assay, and we are one of the rare labs in Africa to have develop this technology. So you could look at here between the ancestral variant and the beta variant. So you talk about neutralization, you could see that people that survivors or people that are vaccinated neutralize better than ancestral variant compared to the beta variant or the delta variant, showing clearly that the ability to neutralize use antibody to neutralize these two variants is more reduced in people that got exposed compared to the ancestral variant. So, and then when we looked at how using serotype neutralization assays again, we look at the ancestral versus beta versus the delta variant, we realize that there's really not really dramatic difference when you come to look at either those that are vaccinated or those that are survivors. And this really good pointer to the fact that there is a need to think about how you want to use this, develop these vaccines. So the other thing that we did, at the time, is basically to use the sequencing platform that we have and the bioinformatic algorithm and machine learning as a way to identify the immunodominant epitopes, and as well as doing a negative selection for antibody dependent enhancers to come up with a library that we actually screen and then selected some vaccine, and then that went into preclinical trials, and then eventually got to clinical trial. This our vaccine after preclinical trial was presented to the Nigerian government as far back as January this year, but apparently there was no traction on that end. And then the vaccine that we're proposing has a broad coronavirus protection. There was no traction from the sense of the Nigerian government, but unfortunately, recently, the Botswana government took over that vaccine, and eventually they're going to be the first African country actually to probably produce a vaccine locally based on based on the vaccine that we developed through this pandemic. So here, for instance, you could look at here, this gives you an idea of how we're able to have the neutralization assay for these vaccines. But what is important about the vaccine that we developed is the fact that it's actually boosting immunity, not only at antibody level, it's also at cellular level. And that really becomes very important, to what we're talking. But more importantly is the fact that I will say we did not need the cold chain for the vaccine that we're developing. Is also dose pairing. We don't need a needle, and it's really intradermal. So it's delivered intradernally. So this whole process that we presented to the government in many ways, has been taken over, as I mentioned, by other government. But in addition to this, we are not keeping the technology to ourselves. We are training people. So, and then recently, we've been able to not only support 14 different African countries to sequence their samples, but we've also been able to train people from those 14 different countries, and then they're using this same platform and in order to respond, to the COVID pandemic in those countries. Just within, we had a 3 weeks training in Nigeria. Within the 3 weeks training, the countries themselves generated about 387, different COVID sequencing. And then they went back home, and then up to now, all of these countries together have generated over 2,500 sequence along with the support that we're providing them. So in cutting this short, we've been able to provide, sequence over 2,500 genomes across Africa, but we also train a critical mass of people, about 1,000. But what is impactful about the training that we provide is the fact that a country like Rwanda, we trained a few of them here, and within a week of going back to their countries, they were able to generate their first genome sequence data. Ghana today is performing genome sequences very well and in one of the top countries in Africa, simply because we trained the Ghanaians how to do it, and then they took the technology and the knowledge back, and then they're doing it very well. Ultimately, our goal is to think about how we can, is actually not to think, is translate, genomic knowledge or the knowledge that we're they're developing to our genomic platform into high throughput genomic, diagnostic and vaccine, tools that can be used, to save and protect our community. And ultimately, we want to use this platform that we'll set up and then use COVID as an example, so that we can use an example to expand the scientific capacity and capability to enable sustainable independent surveillance and outbreak response in Africa. So on this point, I really want to thank this outstanding team of young people working with me, and they're all mostly Nigerians, really working in the trenches to make all this work happen and make this country proud and make this country what it is. But I will end up, also thank, our outstanding partners who work over the years, as well as the general funders who are supporting this program, that we've set up, and that has been very, very impactful, and then it's giving Africa a voice in the community of nations. Over. Thank you.
Yeah, thank you, Professor Happi, for that erudite presentation. Participants, please, if you have any questions or clarifications, just put it up in the chat box. Thank you, Prof, again. It's interesting. We have so many variants now circulating in Nigeria. He said it's about 48 variants, and I wonder if they have the same pathogenicity, as with the common ones like the beta and the delta variants. Yeah, but we'll talk about that after the other presentation. So I want to thank you again for your time and for sharing this very useful knowledge to us. So this will take us to the next presenter. The next presenter is Professor Sunday Omilabu. He's a professor of medical virology at the College of Medicine of the University of Lagos. He's also a hospital virologist at LUTH and a consultant virologist to the Federal Ministry of Health, Nigeria. He's a visiting research fellow to many institutions. He's currently the lead investigator of the Virology Research Unit, and he's the director of the Central Research Laboratory, College of Medicine of the University of Lagos. Prof will be speaking on managing variant-driven waves of COVID-19. And in his presentation, he will speak to us on the effectiveness of current vaccines on COVID-19 variants, the AstraZeneca and the Moderna, and then the long-term implications of the COVID-19 variants. In other words, the immune protection status, the prospect of booster shots, and then improving public health surveillance and response in Nigeria. So ladies and gentlemen, welcome with me, Professor Aremu Omilabu. Prof, over to you.
Thank you very much, Mr. Chair. Good morning, distinguished colleagues. Let me also thank the Academy of Science for giving me this opportunity to come and be part of today's webinar. Let me equally thank Mr. Chair for that brief introduction. 2 months ago, we lost one of our senior colleagues in medical virology, the person of Professor Olufemi Olaleye. So we pray that God continue to comfort the family he left behind and the colleagues. Yeah, the outline of my presentation is as shown. And for viruses, especially RNA viruses, so we are still counting the variants, because as the virus mutates, so the generate variants. But thank goodness, the number of variants that are of concern are very few compared to the mutants that are generated. And the science of generation has to do with the number of infections, number of cases. As you have cases, we tend to have mutations because there are genetic reassortments in the host which the virus passed through. So whether we like it or not, we continue to be talking of mutations, especially for RNA viruses. And coronaviruses are not the exception. But the beauty is that only a small proportion of these mutations impact the transmissibility, antigenicity, the disease severity, interaction with the immune system, and even the detection. So those are those areas of concern with these few degenerate mutants. So the onus is on us not to relax. Let me thank Professor Happi for that presentation. The lab has been doing a lot in terms of genomic surveillance for the country. And that's the way we need to be going to actually really tackle the COVID-19 menace. And of course, we are talking of
Are we losing him?
We lost sound, audio.
The direction of,
okay.
Sorry about that, my interruption from the internet here. Yes, we need to look into our vaccine efficacy studies vis-a-vis the strains, the variants circulating in our environment. That would determine for us the type of vaccines that we would need to give to the populace. Then, of course, we have to look into efficacy of therapeutic management of patients. Then, of course, a lot of gene detection dropouts make nonsense of most of those diagnostics. So we have to look into this area to make sure that we don't miss out cases. And those are the fallout of variants that are being generated. Looking at the structure of the virus, you can see it armed itself with a lot of armaments, right from the surface, then into the nucleic acid itself. The virus has 14 open reading frames, which is very, very strange. The open frames give rooms for a number of manipulations. Presently, they are now dealing with over 27 proteins. And these proteins can do and undo in terms of transmissibility, in terms of detection, in terms of vaccine escape, and what have you. So we really need to be monitoring the genomes of the virus in circulation. You can looking at these three strains of the COVID-19 virus, the alpha variant, the beta variant, as well as the gamma variant. You can see points of mutations is endless. on the genome. It's endless. There are rooms for a lot of deletions, and these are the poser for the future, that we have a lot of mutants lying ahead in future. And the only way to get rid of this is to get engaged in genomic surveillance, just as mentioned by Professor Happi in his presentation. Anything short of this would not go well for us in terms of better management of COVID-19 in our environment. This one is just showing us how the surface protein, the spike, and the receptor using the ACE2, how they congruously align. And of course, with the mutations enable the binding to be more stronger from the spike protein with the ACE2. So there are several mutations on this spike, and that's what actually accounts for the variants that we see. And of course, heightening the transmissibility, affecting our detection. And of course, many of them escape detection using insensitive diagnostics. So we need to really be up and doing. And of course, you can see that the spike protein mutations has made it easy for species barrier to be out of it, because you can see that the virus can move from a number of wild animals as well as the domestic animals to human beings using those mutations. The variants, they've already established that this can be easily overcome. And that's why we're now having a lot of mutants moving around between animals and even humans. So we need a lot of work to do in terms of our surveillance. We need to look into the wildlife, looking into the domestic animals that we keep. They are sources of transmissions, and there are rooms for enabling the viruses to mutate. Like I mentioned earlier on, the more cases you have, the more variants are generated. Just as mentioned earlier on about those variants of interest, so there are so many of them, including the ETA that Professor Happi mentioned. They are of interest, but they were still watching them because they have potentials to develop into high transmissibility, as well of affecting our immune systems. So people are still working on them. We have Kappa. Kappa it has that potential to generate all sorts of phenomena using those mutations to their advantage. The major variants of concern, as we learned earlier on, the alpha, the beta, the delta, the gamma. So they are, of course, what the whole world is grappling with. And of course, delta is the major one that is globally circulating. And then, of course, it is attendance of cases and deaths, even among those that have even received the vaccines. So we'll talk about that later. It's just showing us the distribution of delta globally. You can see that the delta is all over the continents, and we are all witnesses to what is happening globally, including our country, Nigeria, with over 96,000 cases, with over 2,500 deaths. And you can see the numbers that are attributed to the Delta strain. And of course, the Delta, we started picking Delta from around March, around February, March in Nigeria, as shown by earlier data generated. And of course, from our own little studies that we carried out looking at environmental samples, we detected even Delta variants in our environmental water samples in some locations in Lagos State. This is just from the NCDC telling us how Delta has been driving the outbreak in Nigeria in the last couple of months. Just showing the few studies, the result received from the NCDC, showing how Delta is moving around the country. This is our own, some of our own environmental sample studies. It shows that we have several of them Delta since February. We've been picking that since February, but we only recently sequenced them. And of course, we have Kappa. Kappa is among those that we pick from the environmental. So telling us that we now have active community transmission of Delta and some of those variants mentioned. Yes, we can see, comparing those variants, you can see the Delta that has 4 times transmissibility activity compared to other variants. Gamma has around 2 times or 2.5 times transmissibility effect compared to Alpha, which is just about 1.5 or less than 2 times transmissibility power. Then, of course, the Beta variant has a 2 times transmissibility effect compared to the one strain. We can see that the variants are actually trying to actually institute themselves, trying to make sure that they continue to spread without being checked. And of course, tasking our vaccines and of course the developers of the vaccine. So there is that need for us to start looking into tweaking of some of the vaccines to be able to counter those variants that are generated. Of course, we talk of the deletion of these spike protein 6970 amino acids, which are actually is affecting the detection using the diagnostics. So a lot of efforts has to be done to make sure that the diagnostic kits that we use should not be solely a surface antigen detector. So there's need for us to look into using the diagnostic kits that will involve all the major the genomes, the genes of the virus, like the ORF, N protein, envelope protein, RUDRP. So in order not to miss out the circulating variants in our environment. The moment we miss the circulating variants, it does not go well for effective management. Then looking at the vaccine escape and the variants, thank goodness, studies carried out have shown that there is less of vaccine escape with all the variants that we have so far. But they are very weak in terms of curtailing the variants. But thank goodness that the vaccines are still working effectively, trying to modulate or modify those variants. Neutralizing antibodies have shown that so they try to control or reduce the incidence or the impact of the variants. This one is showing that of the Delta strain, using a lot of panel of anti-sera from patients that recovered as well as those that are vaccinated. And you can see that the more exposure an individual has to the virus, the more protective the antibodies will develop. against the variants. Same thing showing a lot of neutralization tests with all the vaccines, the Pfizer, the AstraZeneca, and the Moderna, and showing that the neutralizing antibodies are there. And aside that one, they're talking of the T cells. The T cells are also there, protecting the body against these variants. So not just humoral immunity alone. And for the recommendation, in order for us to be able to move with the virus mutation, so the only word is to start genomic surveillance. So we need to have more of genomic surveillance to actually move with the mutations. Because like I said earlier on, the virus will continue to mutate as long as we continue to have cases. So there will be mutations, and the mutations lead to development of variants. Then of course, this one will enable us to have some regular improvements on our test kits because they have to be monitored. Once we monitor and we know that we have some strains in circulation, so we have to look into our diagnostic kits to make sure that they don't miss out the new variants that are coming out. Then of course, the community health education is very key on the relevance of those countermeasures against SARS-CoV-2 variants. And of course, the non-pharmaceutical countermeasures, masking, hand wash, and of course, avoiding a crowd. They are very, very efficacious in curtailing the virus. It's better not to acquire the virus than to acquire it. So that's the advice. So we should let people know that it's better not to have the virus than to start talking of getting the virus. And then we see most people are going without masks. Most people are still attending parties. Most people don't even have time to do the proper hand wash. All these are very, very efficacious. They've been shown to be those that actually can militate against any of the variants so developed. Our sequencing capacity must be well developed and heightened. It's not just for the day alone. We need to have sequencing hubs in at least in our geopolitical zones of the country to make impact on the monitoring, on timely detection and monitoring of variants. And then of course, the need countrywide and international collaboration among laboratories, clinicians, researchers, and governments to improve our understanding, as well as the translation of results from variants studies. More personalized approach to patient care based on the uniqueness of the individual and the infecting SARS-CoV-2 variant is very, very key in terms of managing patients. So we need to look into the isolate with the type of variant so that we can now match it with more effective or more efficacious management. Then we need more studies to establish the importance of boosting effect or the booster doses and the impact of emergent variants on the establishment of the immunity. So we really need this because the data are not as much, whether we need to have booster doses. And of course, our style of vaccination, we vaccinate, but we don't actually monitor whether they are seroconvert. And then we need those data to show how many actually seroconvert before we talk of booster doses. Not just that they are using booster doses, we should just continue and then join them in using them. We shouldn't do that. So we need to establish their immune status after completing their dose, the two-shot dose. Then of course, looking at the diagnostics is very, very critical. That in order for us not to miss the variants, we really need to look into our algorithm, the diagnostic algorithm, from time to time as a country, so that we don't miss the gene dropouts that are common with SARS-CoV-2 variants. In my conclusion, research, genomic sequence-based surveillance, and collaboration with laboratories and clinicians, they are key to curbing the threat posed by these variant-driven SARS-CoV-2 pandemics. As I mentioned earlier on, the viruses will continue to mutate. So there's nothing we can do about it, just as long as the virus infects an individual. The product coming out from that individual will be different from what actually he acquires. And as the virus moves around, as we have several cases, so are those going to be several mutations. So in my last word, so we need to heighten our genomic surveillance. Thank you very much. Over.
Thank you very, very much, Professor Omilabu, for that wonderful presentation, especially your recommendations, which I can assure you the Nigerian Academy of Science have noted, and we'll see how this can be taken up. So thank you very much. We have eaten into our time for discussion. It's like we have escaped into it, just like the Delta variant has escaped into. Yes. So we're going to look at. There are some questions. There are some clarifications meant for the two presenters. I don't know how you want me to do it. I'm going to go to the Q&A first and read out the questions, and then any of you can handle that. But let me just say that I'm very happy that this has been exposed. It means that we're really busy. We are really busy doing a lot in terms of trying to curtail the COVID-19, contrary to wide views. So my prayer is that we'll try and escalate some of these activities so that people will get to know what is being done, especially in your two institutions. Thank you very much again.
Thank you.
Yeah, so I'll go to the Q&A. Kehinde Ladipo says, Lagos seems clearly to be a focus for the variants, as well as being an entry point to spread to other parts of the country. He's asking, what about Abuja, which is also a major nerve center in the country? Another anonymous attendee is asking, does vaccination after complete recovery from COVID-19 infection offer any additional protection? Another anonymous attendee is thanking Professor Happi for the presentation, and we want to know the state of the rollout of the CRISPR technology for point-of-care testing for COVID-19 in Nigeria. He's asking whether this is available for use nationally and which centers currently have them, as this will help to improve the management of COVID-19. So, Professor Happi, I will hand over the last one to you, which is very important for your brief response. Thank you. Professor Happi?
Yes. Yeah, as I've said in my presentation, so we have the CRISPR-based diagnostics for Lassa and then for COVID-19. As I mentioned before, we have approval for the US FDA. This technology is being used in the US. We had produced some, and then we've done that locally in a few hospitals where we work. That is in Irwa, in Abakaliki, in Owo, and around Ede where we're working. So we're trying to, the problem is the supply chain. We're trying to mass produce and then also make sure that it goes through clinical trial, in Nigeria. Over.
Thank you very much. Thank you very much. Blessing Onyema from the Academy is asking, what is your position about the query of getting vaccinated or not? What are the precautions or steps to be taken before getting vaccinated? And which vaccines? Well, I think for now we have Moderna. The AstraZeneca that is being given, the concentration is on those who have had the first dose. And so they're using that to mop up those group as their second dose. Of course, there are other issues that you need to address there, Professor Patti. Then Aisha Abubakar is, she's happy with Professor Happi. Somebody is happy with Professor Happi. Okay, so which variant would you say is more transmissible in Nigeria? Professor Omilabu.
Thank you, sir. As we know, and as we learned from the presentation, Delta variant is the one driving the outbreak in Nigeria presently. Over 90% of the variants from patients are Delta variants. So it's the one driving the outbreak right now. And it's we can see that it is highly transmissible. And you can see the way it spreads. And like I said, that will also equally help the virus to mutate. As it moves from host to host, then there tend to be changes in the proteins of the progenies. So we should be expecting more of variants. So there are a lot of other mutants, but they are very silent. So that's why we have all those the variants of interest, and then of course the variants of concern. So Delta is the one driving the outbreak now. On the issue of the vaccination, vaccination is the right thing to do if we can give it very early. It is the right thing. But like I said earlier on, we need to not just to give the jab, we need to check whether they actually seroconvert. So that if they don't seroconvert, we can now choose those that will need a boost. It's normal that we don't respond the same way to antigens. So we need to check among those that have taken the jabs, whether they do seroconvert, and then what's the status of that seroconversion? Do they have enough log neutralizing index in their plasma? So we need to check all these to make sure that they are fully prepared in case they come in contact with wild strains or the variants. And of course, as we can see, so there is still need to look into some of the vaccines. So as some of the manufacturers are doing right away, they are tweaking their vaccines in consonance with some of the variants of concern that are in circulation. So those are my response to that. Thank you.
Yeah, thank you very much. Why is it? Is there any particular reason why there is clustering in the south of the variants?
The clustering is
from the map. Sure, there seems to be clustering.
The clustering is actually over Nigeria. It's all over the country, but there is more of testing in the south compared to there is more of testing.
Okay.
So that's what that is showing. Thank you.
Okay, that's fine. What's your take on mixing vaccines? Do you think it provides better immunity?
There is few data on this. From the few data available, they show that it shows some promise in terms of boosting the immune response. But we still need to have more of data before we go into conclusion.
Okay, so thank you very much. I think we have some questions on the chat box. Let's see. Please, if you can respond to any of the, if you go to Q&A, I mean, this profs, the presenters, if you can respond directly, just type your answer if you have any quick answer to those questions that have been raised in Q&A, because we're running out of time. Yeah, okay. I find it exciting at a time when many, okay. Yes, so yeah, Kalu Onoha is making a very important statement here. He's happy for the excellent presentation. It's great to know about the kind of work that is being done in this area right here in Nigeria. We find it exciting at a time when many people see only the wrong things going on in Nigeria. I agree with that. Professor Happi, can you shed more light on the variants of concern, of interest, and various? Okay, I think this has been dealt with. These ones are the alpha, beta, delta, and gamma.
No, but I think the question is, do you, more light on variants of concerns and or
yes.
of interest?
Yes,
and they're under investigation. Generally, in the area of genomics, we call a variant of concern when the variants have been shown mutation that are associated with increased transmissibility or increased pathogenicity or death. Then that becomes a variant of concern. When you have a variant of interest, that variant of interest is a variant that shares some mutation with variants of concern, but they have not been demonstrated to be associated with increased transmissibility or increased death. Now, variant under investigation are variant that show some mutation, with either variant under of interest or concern, and then they are being monitored because there is a potential for it to evolve over time and become either a variant of interest or a variant of concern. So that's how we categorize them, because they definitely should be sharing mutations that are actually located in the site that is very, especially in the spike protein, for instance. If you have mutations associated with spike protein in areas that are involved into either facilitating increasing the ability of the cell to penetrate the cell, the host cell, then you become a variant of concern. But then once you show some of those mutations, depending on where the mutation is located, you could be classified either as a variant of interest or a variant under investigation.
Thank you very much. Just still on that, Professor Nasidi wants a confirmation, more for you to throw more light on that first case that caused by the beta variant that was detected in Nigeria. Do you have any information on what proportion of COVID-19 in Nigeria that is currently caused by these beta variants?
Well, it is clear that most of the infections are driven by variants of concerns, and those could be either alpha, they could be delta, and then they could also be the Eta variant. The Eta variant actually seems to be the most dominant variant in the country, and the second most dominant in Africa. Based on the study that we've done across the continent after the Beta variant. We haven't really established in the case where most likely because of samples are biased, because we sequence mostly from samples that are in the south and in the southeast and the southwest. It is very possible that a lot of those that we may have the beta variant, but that has not been demonstrated. We haven't demonstrated that clearly here. Neighboring countries like Cameroon, where we have sequence, we've identified the alpha, the beta, and then the delta variant in some countries around Africa. But in our case, because of the bias, for instance, we know that some of those variants could be. And then, by the way, we just had a new finding. We just completed sequencing sample from a country, neighboring West Africa, neighboring country like Burkina Faso. And we found that, the Delta variant that was reported in India around September last year seems to have been circulating in the Burkina Faso area of West Africa much more earlier than it was in India. So it is something that we're really looking at now, and then to see exactly whether there could be a potential, whether they are, related, whether the discovery of this new variant in Africa was related to travel history or is something that has been circulating in Africa for some time. Over.
Thank you very much, Professor Happi. Let me agree with the president of the academy, Professor ECABRED, FAS, that so much work is going on in Nigeria, but the problem is lack of traction from governments, and as you, Professor Happi, put it, so we need private sector intervention. I mean, we can't overstress this. Emeritus Professor Augustine Esebwo, FAS, is very happy with the presentations from both professors, and is very proud of the work that is going on in those labs. Now, his question is about data, sufficiency, and quality, which will enable dependent policy formation from a statistical point of view, especially against the backdrop of the classical assumption of normal distribution in these studies. Do you have any comments on this? Either of you?
My own brief comment will address the availability of the state-of-the-art facilities to actually carry out the sequencing. Until now, it's only Professor Happi's lab that we have NGS. But thank goodness, in the last couple of weeks, NIMA has gotten one, and Lagos State Biobank has also gotten another one. So that will actually increase the data generated on those the variants circulating in the country. Especially, we started with that of the Lagos. That's how we're able to provide information on some of those samples we picked from environmental samples. And we were able to show that the Delta has been in the country since February. And you can know the implication of that by the time we start talking of the incidents about a month ago. So with the availability of state-of-the-art facilities and of course the support from our government, and like Mr. Chair mentioned, the private individuals too needs to come in so that we'll have funds to actually drive these sequencing studies. So more data will be coming because we have plans to look into all those that are positive. And like I mentioned in my presentation, if you want to win the war, we have to know the variants that are actually circulating in any environment so that we can prefer necessary and adequate countermeasures against it. And of course, we shouldn't even miss any of these isolates. So that's why it is imperative that we need sequencers and then, of course, funding to make available all those consumables actually required for operating NGS. Thank you. Over.
Thank you very much.
I don't think that, personally, I don't think that the data, yes, the data is a bit skewed, but the data that we generated, as I mentioned in my talk, we've done 28 out of 37 states in Nigeria. That's significant, and that covers quite a number of states in the country, even though these are not in real time. But I think that if effort can be put into the area of collecting data, collecting samples, and then send them to the sequencing centers in real time, then we should be able to have a better understanding of what is circulating. But at least we've covered more than 2/3 of the country, sequencing data, sample from more than 2/3 of the country. And that gave us a good sense of what is going on. I think the problem is more or less just how to coordinate and then get the samples in places whereby we could generate the sequences. I think the problem is not the sequences, but I think it's how do we coordinate and then collect samples in a way that we can have a true picture, representative pictures of Nigeria as often as possible when it comes to determining the level of variance circulating in the country. Over.
Thank you very much, Professor Happi. I'm also happy the way you're also building capacity for this area of specialties. Okay, yeah, your PhD students, your master's students, this will make us to have a critical mass of scientists in this area, which will help us to generate, conduct research, generate evidence for policy and practice. Thank you very much again. Someone is asking, Saludin is asking, if there's any measure put in place to prevent another possible outbreak of infection due to large number of variants being currently detected across the country. I think the second presenter dwelt on this in his recommendation that we should concentrate, continue to the MPIs and the non-pharmaceutical intervention, put on your face mask, wash your hands properly, maintain distance, social distancing and all that. So I guess it's the same measures. All we need is just to continue this. Presently, people have relaxed, and I guess that's why the number is going up. Thank you very much. Okay, so okay, I think we are, I'm most happy for practical experience you shared. Okay. My question to you is on the fact that mutations of this virus do happen most oftenly in immune compromised persons. For example, people with HIV/AIDS, etc. We have large volume in Africa. What do you say to that?
Yeah, it is true that, you know, if we look at a historic medical background of most patients where we've seen this variant emerging, you always realize that there is some underlying issues, and people that are immunocompromised are often, you know, are the ones driving this process. I think for most of the data that we've generated from the sequences that we've done across the country by the time we start, not only just within Nigeria, but also across Africa. When you look at the data and then you go back and dig a little bit deeper into the clinical profile of the person, then you realize that a lot of them, Professor Nasidi is absolutely right. You know, people that are immunocompromised, not only HIV/AIDS, but other underlying conditions are the one driving this. And the same, I mean, same for people that, I mean, for people, I mean, with vaccine, you know, breakthroughs. Over.
Thank you very much. I think both presenters have made a point that we need more sequencing facilities across the country, at least one in each geopolitical zone. This will help to also offload decrease the pressure that is currently being faced by very few facilities. So this is one thing that the National Academy of Science should be able to advocate for. So thank you very much. I think we are on course now. Let me see if we have any other. Someone said he has taken AstraZeneca and Pfizer, and he's willing to donate his serum for, what is he saying? Yeah. And yeah, he's willing to provide the samples to show the level of seroconversion. Well, okay, thank you very much for that.
We'll be glad to have that one if you can bring to us in Lagos State Biobank.
Yes.
We have the facilities to check the immune status.
That's good. So,
yeah, but I do think that these are kind of studies that we want to be seen happening in the country, and
exactly, because this is just one person, you know.
It's one person. As I mentioned, we showed in our data, when you look at people that are naturally, you know, survivors, those are people that got infected and those with vaccinations. You know, we did that study already in a good number of people across, you know, the southwest and southeastern part of Nigeria. So you see really no difference in terms of antibody neutralization between somebody that survived natural infection and those that are vaccinated. It shows that actually vaccines actually provide also a good, you know, a good level of protection afterwards. I mean, as well as people that have survived and have been infected with COVID. I think it's very good, it's very promising, you know, to see that happening in the people, I mean, across the region. Over.
Thank you, Professor Happi. Have you reported any mixed infection in your work? Any mixed variant infection?
No, I think I saw that question from Professor Iruka Keke.
Yes, Professor Iruka asked that. Yes,
it's very rare. We've been looking out for it because it's a very rare event that you're really looking forward to, but we've not seen so far. Over.
Yeah. Okay. Thank you very much. Emeritus Professor Augustine has a second question. He says, at what point will it be at what point will we be positioned to lead in the production of a variant of interest and concern for other researchers elsewhere to follow? When you say we need more data, do you have a priori a calculated volume of data needed for reasonably reliable conclusions on your studies? So he's hitting some statistical questions. Do you have any comment on this? Any of you?
Well, I do think that, you know, it's going to be very difficult if you're looking at the population, a country like Nigeria, and then if you want to calculate, I mean, it's going to take a lot. And it's also very difficult to predict exactly how you want to do it, because this is a very fluid and dynamic, you know, pathogen, a pathogen that just keeps changing. And by the way, when we talk about 45 different variants, 48 different variants that we mentioned, so these are variants that, you know, keep, I mean, as time goes on, you just keep seeing these numbers. They are not static. But then the variant that we observe, as we mentioned, you know, you have variant, I mean, you have all these 48 variants, but out of the variant, we found only very few, 3 or 4 of them that fall within the category of variants of concern, variants of interest, and under investigation. So it gets very challenging to calculate and then say, okay, this is a number that I will need exactly to make any meaningful conclusion. But I think what we do generally for genomic data is actually to look at a percentage. If you're looking in a given population and then track over time, when you reach a certain threshold, then you can start making, you know, that assumption that something is taken over. But I mean, it's very difficult to answer that question, sir. Thanks.
Okay, thank you very much. Do you think there's any need for a graduated lockdown as a result of this explosion of the new variants?
I guess, you know, a lot of these things, as Professor Omilabu alluded to, I think you can lock down as much as you can, but if people refuse to abide to the normal, to the basic safety, you know, regulations, I don't think that's going to help. I think the whole idea is actually to how do we bring down transmission to a level whereby it is bearable, because it's always a function of viral biomass that is available out there. So if you bring down the level, the biomass down to a level, you know, that is understandable, eventually, you know, this will fizzle out. I think it's, how much do you lock down, and then for how long do you lock down? We've seen countries that have locked down. You look at, New Zealand and Australia, it's backfiring because, you know, you need to balance these two effects. You need to balance the economy, you need to balance health, you need to balance mental health, and so many things. So it's very difficult to think about it, but I do think that if we can bring some measure of saying you want to lock down, but that has to be accompanied with some very strict measures in terms of respecting, you know, some basic, you know, safety and hygiene procedures, and that really is what we need.
Okay, thank you very much, Professor Happi and Professor Omilabu, for the wonderful presentation and exposures. You know, a lot of us have benefited from your presentations, and more importantly, you have proven that we are busy trying to do something on this in Nigeria, contrary to comments and you know, what people say. So at least in Nigeria, we have good people like you, scientists like you, that are producing this evidence for policy and practice. I want to thank you. Like I said earlier, it is important you escalate some of these findings so that people will know what you're doing. We need more facilities, you know, in various areas, and then and then it's also important that we use what we are getting to inform policy and change practice. Thank you very much. I want to thank
you, Prof.
The participants. We hit 76, about 76 of us here. So this is a very huge success. This is a very important one. So I want to thank all the participants for their patience. So I'm going to call on Professor Konji.
I'm sorry, sir. Just before you do, I wanted to, please pardon my misbehavior, just to ask the experts to do something that they probably don't like to do, which is to kind of predict where is this going. Are we with the variants, with the non-pharmaceutical interventions or not? With the natural history of the virus, supposedly, where do you think this is going? Are we going to where to fizzle out, or we're going to where we'll be getting booster shots every year?
That's a tricky one.
Very, 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 very tricky. Let me just start by saying that the virus is also not resting. All the virus is looking for a host to actually sustain the virus in circulation. And as long as people don't abide by simple non-pharmaceutical countermeasures, once the virus finds place to germinate, so the story will continue. The best way is not to have the virus at all. And once we can do that one, the virus will not find a host to germinate, and the numbers will be going down. As long as the virus finds hosts, it will continue to mutate. That's the fact. So we should try as much as possible. All of us should cooperate and fight the virus by reducing the number in circulation. That's the only way. No matter how much we lock down, you can see from the data shown by Professor Happi, despite our 2 lockdowns, there was still movement of the virus at night, as we know.
Yes.
So what are we talking about? So it lies on the people. If they want the virus to fizzle out, so they have to all put our hands on the same deck to control it. Over.
Thank you very, very much for that response. Yeah, so Executive Secretary, do we have Professor Konje on this platform? Oh,
yes, please. Thank you very much.
Thank you, Prof. Yes, so vote of thanks, yeah.
Thank you. On behalf of the President of the Nigerian Academy of Science, Professor Ekabret, FAS, I want to thank most sincerely Professor Benjamin Uzochukwu, FAS, for expertly moderating this webinar. We thank you very much, sir. I also want to thank the presenters, Professor Christian Happi of Redeemers University and Professor Sunday Omilabu from Luth for very stimulating and educative presentations. In fact, there was a time I was so scared when they said we have 45 variants from me about in Nigeria. I want to thank both presenters. They have made our day. I want to thank all attendees. Mr. Chairman, sir, at the peak period, we had 79 participants. I want to thank you for the interest that has been shown and by the questions that came. Lastly, I want to thank the Secretariat of Nigerian Academy of Science under the able leadership of our ES, Dr. Odubanjo, for always putting programs together that will educate all of us. Mr. Chairman, sir, I have requests from a large number of participants that the slides of the 2 presentations should be made available on the website of the Academy. I want to thank you, sir, for the way you moderated and for a very wonderful day. Thank you very much, sir.
Thank you very much. Madam President, Professor Ekabret, all the fellows on this platform, the Executive Secretary, our panelists, thank you very much for your participation. At this point in time, we will close this webinar, and then I'm hoping that in no distant future, the National Academy will call us up again for another webinar. Before then, it will be important to look at some of the recommendations that have come out from this webinar so that we'll see how we can take this up for policy and practice. Thank you very much. And goodbye.
Thank you. Thank you, Chairman. Thank you, everyone. Have a nice day.
All right.